So welcome to video 6 in our course B07, our introductory video looking at accessing databases in R. So there's lots of examples, and we're just going to go through a few different examples here for how we can access database data. An example here is modeling COVID regional data with R. So this loads packages to get worldwide time series data by country, sourced from the WHO, on the frequency of cases and deaths and so on per day across different countries. This is a lot of data, and it's all available on the WHO website, and we use R to access this. We're using a package as an interface for this because all the functions to access this data is there. So this is an illustration of how we can access available data from a website. So we have to install this package COVID regional data, activate it, and then set up this link to get national data. So this kind of sends our national data to this variable called NOTS. This might take a minute to download that information, and you might find that is very, very large, might even be larger than this value now. What that is looking at, it's 262,000, because I looked at it in the file, and if you use Ireland as an example, every country is in there. You'll see at some point the first column denoting the time, then the location, then the number of new cases, cumulative cases, and new deaths and cumulative deaths. But this is 262,000 rows. So it's a very, very large data set. Well, we need to look at it as well. We're going to use some of the packages we know already: dplyr, ggplot, another one called scales, as advised by the package instructions to open it. What we're going to do is take a few countries that are of interest after we install the packages, like Ireland, the UK, and Germany. There's many other examples that you can try some yourselves. We're going to get national data for those countries only, NX1, without their verbosity. If you want, you can turn verbosity on or off, up to you. We can check the structure to see how many rows are present. And then we can take our X1 NOTS values, and pipe it out to ggplot, and we can plot the deaths over time. So we can look at the aesthetics, and we're going to look at the date and the deaths new. So if you see here in the previous columns, we had those variables present, and we've got a column is the country. We're going to add a line, a geometric line with a kind of opacity level of 0.4. So if you make it, you can change alpha to whatever value you want from 0 to 1. And then we're going to add in the date and the reported COVID-19 deaths on top. We're going to scale the y-axis so the labels are comma, and then we're going to add on the legend at the top, and then use the column guide for the country. Now, I didn't write this code myself. This is from the package, but you can use code in packages to make some nice plots. If you see it there and it's useful, you can use it. And you can use plots like this, where you're looking at Germany in kind of mauve, Ireland in green, UK in blue, and you see the reported deaths over time, where the UK and Germany are higher than Ireland, obviously because of the countryside. So you can think of factors over adjusting for the size of the country and things like that as well. So you can see how there's kind of different rates there. Another quite different example of accessing databases is for virus sequences. So you might want to examine some sequences. It could be viral ones, they could be any type. You need to know the accession numbers in this example already. And we do. We're looking at some partial VP1 sequences from foot and mouth disease viruses, FMDV, for the serotype SAT2. These are just chosen for illustration, and we just have 1, 2, 3, 4, 5, 6, 7 of them there only. So you put these in a vector called list1. So what we can do is take that list1 set of sequences, refer to the sequence name as being inside list1, ask for the species name and make sure that our sequences are read as characters, and use this function read.genbank and download those sequences from GenBank. So you can ask, what did I download, just to check if it worked. We can check the sample names or the sample labels for our sequences and look at the attributes that they have. That will then allow us to do further things. And when we find this out, we'll find that we have these sequences here that are downloaded, and they're all foot-and-mouth disease virus SAT2. So next, we can do things like take our sequences that we have just read in here. We can do a multiple sequence alignment and call that sequence aligned. And here we're going to use muscle instead for this particular alignment. That's muscle version 3.8.31. Just for illustration, we then can view our sequences as before. So we actually reuse our code from before and put it in here, really. In the same way, we can convert our multiple sequence alignment from seek aligned down here, using sequenor again, put that into a distance matrix called myA, get our distance alignment again called seek distance. We can then view it. So we're rounding the numbers to just 2 digits and then viewing it as a matrix. So that will give us something like this. So you can see there the relative distances between these set of 7 sequences. If we look at this then further, we can write out these DNA sequences by giving it a file in the format FASTA. We're not appending it, we're writing out a new file. I've set the column width to be 10, but the column separator is 0, so it's just a single FASTA type file. And we can also do things like use the nj function in ape we had before to plot our phylogeny 2. So if we do that, we'll get a phylogeny something like this. So you can see the relative relationships of these different 7 viral segments. You can even try yourselves to add on a scale bar, and we'll do that in the labs itself. That's the number of substitutions per site to give a sense of how distant they are from each other. A final option within this whole area is to query a database looking at a bunch of sequences related to a species. Here we're going to use the VP1 gene region as an example for foot and mouth. But the same kind of procedure could be done for many different species. We know in advance that we're looking for something in foot and mouth. We're just querying the database to see what's out there. So what we're doing is you're using the Entree database. We're looking at the nucleotide database. And our search term is foot and mouth disease virus as the organism. And we said that we're returning a maximum of 10,000 or so sequences. And then we're going to check on our search what do we get at the end. So you might find there's actually a lot of sequences that are returned. So if we look at that, we can look at the search one accession IDs if we want. And then we can even say, using this function onto fetch to say in the nucleotide database for these IDs, which could be numerous, could be very few or numerous, return the FASTA sequences from them. We put them into sequences.for, seek4, check that it worked, and then write that into file if you want to do so. So that's why we can ask, I want all these sequences. It could be GenBank, FASTA, whatever format, and we can download them all. To have a more specific term, because there's a lot of having a bunch of sequences doesn't mean that they're related necessarily. We could focus our term on the VP1 region only using a Boolean term. So we're going to say we want the VP1 gene as well as the foot and mouth organism. So you can see that down here. We've got foot and mouth organism as the organism, and we're saying and as a Boolean, the VP1 as the gene. So you can see this is all in double quotes. So this is effectively a search term sent to the website on NCBI, returning an effectively unlimited number of entries. And then we can look at this further. So you got search 2 here. What we can do is examine them further. For instance, in the demonstration video, we'll look at how we can, for example, take that set of sequences, align them using plus null omega because they're missing data, so you don't want to use muscle. We can then take sequence aligned 5 here, send it to convert it to a sequenor alignment, then take my A5 and create a distance matrix out of that. And then we can plot using a neighbor joining like algorithm in APE to construct a fan-shaped tree here. So we're showing a fan shape because there's a lot of sequences here. So if you did it with other methods, it may not be as clear. You can see there the relative relationships, and you can see that the tips are not actually shown here because we turn the show tip label to be false or off. So there we can see our tree, and we can kind of develop that further after this basic tree. So this kind of illustrates how you can use traditional search terms to query a bunch of sequences, all that are of interest, and construct a phylogeny from them to at least get a sense of are they all actually VP1 regions or not. Our final trick we're going to learn is to deal with FASTQ files. So there's a test FASTQ file in the N-Drive folder, Genomics Public Omics Learning, where you've got test1.fastq, which just contains the first 1,000 lines of a FASTQ library in the forward reads. It's of a foot-and-mouth disease virus, and we're going to use as a library called short-read to do some functions, and we're going to plot some things with ggplot as well, so you need to install them. What we're doing here is sending our function, our FASTQ, to a function called readFASTQ, and we're assigning that to a variable called FASTQ1. This is an object containing the FASTQ file in essence, and we can look at the DNA sequences that are inside from it. We can get the quality scores from the FASTQ and look at it as a matrix, and look at plot those quality scores as a particular value numq scores, and then we can send numq scores to get the row means of that value and plot that as a data frame in our average scores. So we have stored the average scores for each read and made a data frame from that. So we have the data frame here, so plotting that down in ggplot here for the average scores per read, and we're adding a histogram onto that of that data. So we're just simply taking the average score and plotting the histogram of that, which means they're plotting the frequency on the y-axis for each read, and then the average score of that read. So when we do that, we'll get a plot like this. So what you see is the frequency or the count, the number of reads corresponding to each interval, and that ranges from a lower average score of maybe, I don't know, 17 there, all the way up to maybe 39 over here. And this is the average score here across all the read. So the individual bases may have differing quality scores, where we're averaging them for the reads. And what you see is there's this kind of skewed distribution here, where the mode of maybe 38, 39, and then a median maybe close to 34 or so. And you should be aware that these average quality scores correspond to the Fred's quality scores here, where we take this value here, we divide by 10 to get a value of 2, and then we take simply 10 to the minus the power, negative power of that value. So we've got 10 to the minus 2 corresponds to a probability of an error of 0.01. Likewise, 10 to the power of minus 3 corresponds to probability of error of 0.01, and so on all the way through. So you can see a higher quality score corresponds to a log-based transform of the probability of an error per base in the read. So this is an illustration of how we can use certain libraries like short-read to access information about FASTQ files and plot it in a very code-efficient manner. And there's lots and lots of other things that you can try. This is just an illustration. So what we've looked at here is extracting CSV data, large-scale CSV data from websites, how we can download, take existing accession numbers and download them and look at them further. How we can do search terms with inside OR code and retrieve that data. And then finally, the fourth item here is that we can take, say, for example, a FASTQ file and also assess that with OR. So these are all different ways in which you can access database data or use code to understand large-scale data more efficiently. So that concludes our introductory videos on this area, but please see the demonstration videos, which will give details on code in much more detail.